All right. Okay. All right, we're back. Thanks, guys. All right. Thank you, Cal. The joys of live streaming and being tied to frame rates and the modem. So thanks again, everyone, for coming. I know this audience understands biology really well, but in case you don't, or this is something that's kind of new to you, I just wanted to start by saying a little something around biology. Biology really is technology. I think that's something that's new for a lot of people. Biology is not just a field of study. It's actually an applicable technology to change the lives of people like you and me. And this technology is getting faster, cheaper, and more powerful every single day. What used to take, back when I did my degree, a full day to do a hand PCR can now be done extremely quickly, and in massively multiplex. So these types of tools and the further digitization of biology is driving the ability to do things in ways and in speeds that were unheard of before. So at IndieBio, what we did was we built an accelerator to help scientists with a unique insight be able to apply that insight and build a product and a business that would be revolutionary and solve what we call intractable problems. Problems that seem to have no possible solution, but with the ability to actually manipulate the biology itself, that solution becomes solved. So in doing so, over the past couple of years, we at IndieBio have actually become the world's largest seed biotech investor by volume. This is from CB Insights, and what this actually means is we're learning. We're learning very quickly across a broad spectrum of types of biology, from therapeutics to food and ag and many others. And we don't just do this alone in San Francisco. We have global support networks beyond just our accelerator. And we've built relationships with corporate giants like BD, Unilever, and others. And this is to help shorten the distance between scientists walking in, trying to build a business, and their customer, which is always the hardest part. How do you find someone who actually wants to buy something at industrial scale or things like that? So this is what we're doing this for. We also have academic relationships. We've gone out and spoken with and held events with universities around the country, from Harvard to Scripps to Berkeley, down to Stanford. All of these events have been around entrepreneurship and how to turn insights into revolutionary businesses. And finally, we're starting to see global relationships emerge. Relationships with actual nations. Governments have come and wondered and asked, how can we take this model and apply it in our home country and accelerate healthcare and biology there? We really care about this industry. That's why you guys are invited here in this room. This industry, biotech, means a lot to us, and we think it's changing a lot. For us, we're excited to say that batch 1 and 2 are thriving. We've graduated 26 companies total in the last 2 batches, and they've raised in excess of $23 million alone in the last year. Batch 3 that you're about to see has some amazing companies as well. Like I said, from 21st century medicine to new biotools, future food and agriculture, and consumer biotech. I'd like to introduce the team that makes this all happen. I'm Arvind Gupta, Managing Director of IndieBio. This is Ron Shigeta, the Chief Science Officer. Ryan Betancourt, Program Director, and Alex Capelliman, our new Program Manager. So I'd like to hand the mic over to Ron and Ryan, who are going to explain a little bit more about how do we actually do this? How are we accelerating science? How are we accelerating the building of these businesses? But first, I have to do a quick live stream disclaimer. This is, sorry guys, lawyer stuff. Lawyer called me and told me that I had to do this. This is not a general solicitation to the live stream audience. This is an offering really only to the 400 accredited investors that are sitting in this room today. So thank you all for that. All right, Ron.
Thanks, Arvind. All right, so one of the things you'll notice, and one of the reputational sort of like big points for IndieBio, is that all the presentations you'll see today, all of the 14 companies and 15 companies that you're going to talk about, they've all achieved some form of prototype or proven their principles commercially viable somehow, right? So there are no vaporware talks. And so one of the big points that we take very seriously is we want translation from bench to market. We want a viable product that comes out of their time here and out of the program. And we do this in a 4-month period. Our program's 4 months long. One of the reasons we have this is because we have a lab on site. It's a very dense environment. The science and technology are integral with the program every day of the week for 4 months. In this class, the class has gone in and they transformed the lab to their needs. We've incorporated plant technology of greenhouses in the lab this time. Lots of food prototyping as usual. And fermentation technology. But that's versatility in the lab is one thing. But by far the biggest jump for the companies and the entrepreneurs that are in the class is about learning about product-directed discovery. How to do science to discover how a product can actually become viable, how you can make something that people want. This is three generations of prototype from one of the companies. That is a pencil there and some tape. It's really about using whatever you can to make sure that this is something that really works in the hands of others. Food prototypes again, loads and loads of them in the fridge. Oh, and then most importantly, demoing often. Taking those prototypes and showing them to people, showing them to customers, showing them to the mentors, getting feedback immediately, and doing many, many cycles of this as we compress the cycle of work so that this can happen in the period of time that we have. We also use a large network of the facilities and collaborations with institutions in the area. The Bay Area is very lucky, very rich to have all of these different institutions that we work with. They provide equipment that we don't have if the companies need them, and really all their expertise. All of these things are very, very important to the success of science. So with that, I'm going to turn over to Ryan, and we'll talk a little bit business. There you go.
Thank you, Ron. So welcome everyone to the IndieBio Demo Day. So we take a bunch of companies in 4 months and they come out and they have sales, they have partners, they've raised money. How do we do that? That's the question we get asked all the time, right? It was previously impossible until we started it. Now it's possible. We do it on a 6-monthly cycle. So how do we close the distance between science and making money, right? That's one of our main focuses here at IndieBio. We start first with the moment the companies and the founders come in, they are already outside of their comfort zone. So many of these founders have actually come from other geographies. Most actually don't come from the San Francisco Bay Area. They come from geographies and countries where English isn't even necessarily the first language spoken. So from day one, they're innovating and building businesses in a new world for them. They focus on going hard, and we push them hard from day one. That means cold calling, right? That's scientists who actually pick up the phone, cold call, even if they choke, and they do it again and again and again, and that's how we build entrepreneurs. We focus on not the first cold call, not the 4th or 5th, but when you close, right? So this is outside of, this is not just getting an LOI, this is building your business. We focus on time to revenue, right? So that's the question we ask every one of our founders. How long until you have a product which makes money, right? Day one, right? That is a question that many, especially those that come from academia, are unused to hearing. They have 4 months. The clock starts immediately. We focus on knowing your numbers. Everyone at IndieBio knows their numbers. So through the boot camp, which we call IndieBio, the focus is what's your market size? How do you get real product market fit? The market teaches those entrepreneurs. We start from day one with interacting with the market. That means going out and closing customers, right? And so that's market size, that's cost of goods sold, that is what is your profit margin, right? What's your vision for the next 10 years? How do you get there? Let's start there and work backwards. And then finally, we're building real companies. These are not theoretical businesses that we build in business school. These are real companies. So we teach founders how to hire, for strength and also for weakness, and also how to hire and fire. Right? All of our founders and all of our companies have hired during their duration, from scientists to salespeople to marketing to PR to technicians that help them out in the lab. So I'd like to kind of push us into the next segment, which is coming up. A quote by one of my favorite theoretical physicists, Freeman Dyson. Many of you are probably familiar with Dyson spheres. So one of his quotes is, now after 3 billion years, the Darwinian interlude is over. Humanity now has the tools that we need to manipulate and use biology as a tool of creation. And so now I'd like to welcome you to the post-Darwin world and welcome our next 15 batch of incredible world-changing companies. Thank you.
Nice job. Hi, I'm Sophia Wang. I am the CEO of Mica Works. Mica Works is a biomaterials company that is changing the way things are made. And we're starting today with leather. Leather's been around a long time. It's beautiful, durable, functional. It's primal, it's a hide, it feels good to the touch. It delivers status, luxury, and comfort. And we love it because it's natural. But leather is a co-product of the livestock industry. Think of the resources and time this takes. This industry uses 50% of this country's water supply and 30% of the Earth's land mass. This industry produces 18% of the Earth's greenhouse gas emissions. And producing leather is costly and wasteful. Only 30% of an untreated hide gets turned into leather. The rest gets thrown away. Tanneries produce toxic waste, subject to regulatory fines and human health violations. And because leather is tied to livestock, price and quality are vulnerable to climate and availability. But people love leather. Cattle populations grow with human populations at 3% a year, but the global leather goods market and consumer demand is growing at a rate of 4 to 7% a year. So desire and demand aren't going anywhere, and neither are the problems. This is an opportunity. Mica Works has an amazing answer, and it comes, of course, from nature. Our technology is based on mushrooms, one of the most abundant resources on the planet. The root fibers of mushrooms are called mycelium, and it's a naturally strong fiber that grows on almost anything under the sun. We have discovered a way to turn mycelium and agricultural waste into a natural alternative to leather. So we're not replacing leather, we have something even better. People are always blown away by how much our material feels like leather. That's because, like leather, it's a skin, it was grown, it was alive, it's organic. It's also durable. It holds up to industrial sewing and fabrication, and our mechanical tests show that our materials outperform deer and lamb's leather and synthetic leather in tensile strength tests. It's versatile. We've created a nearly endless range of colors and finishes, and it's engineerable. So we can customize growth to express patterns and textures. We can customize for thickness, density, and three-dimensional features. You can't do this with animals. You also can't grow an animal to create a hide of any size and shape. We can do this with our technology. When we first started at IndieBio, we had grown a piece that was 1 square inch. Within a month, we had grown a sheet that was 2 by 3 feet. Within 2 months, we had grown a piece the size of a full-scale cowhide. That's 1 cow saved. And we're quickly scaling beyond that. So we're scaling fast. We can do that because our process is rapid. Compare the 2 weeks it takes us right now to grow a sheet of our leather to the average 3 years it takes to raise an animal to maturity to harvest a hide. So this is an ultra-sustainable technology with no downsides. Our closed-loop process has low water and energy requirements. We can grow multiple sheets on the same ag waste, which means this is a carbon-negative process. And at the end of product life, our leather is biodegradable, and mycelium is a benefit to the earth, not a pollutant. Best of all, our low-complexity process means that we can compete with leather right now. In our first year of pilot production, we'll be making sheets of our leather at $50 a square foot. We can compete with exotic leathers used by manufacturers of high-end footwear. That's our first target market. With greater volumes, we'll be able to bring the cost down to $30 a square foot. We'll compete with top quality leathers and serve manufacturers of premium footwear, apparel, and accessories. And within 4 years, we're going to bring our raw material production in-house, which will drop our costs down to $5 a square foot, which means we can compete with nearly any leather on the market. We'll serve manufacturers of mass market leather goods. We know we can scale with these projections because we're modeling our scale up on systems already in place to produce edible mushrooms in commercial volumes. So we'll use similar tech and reorient it towards making materials. We know we can make 150,000 square feet of this leather in a 6,000 square foot facility. So we already know what we need because this infrastructure already exists. We'll be competing with natural leather, conventional leather, synthetic leather, and biofabricated leather. But unlike us, these competitors do not have a solution that's scalable, sustainable, animal-free, and low-cost right now. The markets are huge. Of the 23 billion square feet of leather produced every year, the largest outlet for that is footwear, valued at $47 billion. The leather goods market is valued at $200 billion, and footwear represents almost 60% of that market. We've got great traction. I can't disclose names, but we're working with 2 global footwear manufacturers and a global apparel manufacturer, all household names, to get our leather into products next year. Our patent's been granted. We've filed another one for this tech. We'll have a full product data sheet and a pilot facility by the end of the year. Pilot production starts next year. We'll scale to commercial volumes in the following year, and that'll allow us to expand beyond footwear to other leather goods markets. We have an amazing production research team, and we are so proud to be working with our advisors, Dr. Drew Endy, co-founder of the BioBricks Foundation, Dr. Amanda Parks of Manufacture New York, who shares our commitment to innovating fashion with technology, and Kevin Bayek of Lift Economy, who shares our commitment to social impact and sustainability. Our CTO, Phil Ross, invented the field of mycotexture, and he has 20 years' experience developing mycelium as a structural material. Our senior advisor, Eddie Pavlou, has 20 years of executive management experience. And I started working with Phil 10 years ago when no one had heard of mycotexture. And I'm so proud to have helped bring this tech to where it is today. Mushrooms, we've known their culinary and medicinal value for a long time. And we're only now starting to unleash their potential as a structural material. Mushroom materials are now being used in packaging, furniture, interior design, and more. And we are so excited to be bringing mushroom leather to the world. We're already making product. This is just the start. There's so many industries that we're going to change for the better. As one of our partners likes to say, we are on the cusp of the mycocultural revolution. We are raising a 2.3 million seed to scale to pilot and grow our team. Our round is firming up, so please get in touch to help bring this amazing technology to the world. And please come see us after the presentations and see and feel for yourself what we've made. Thank you.
It's on? Great. Hello, everyone. I'm Maria Soloveichik. I'm the CEO of Synthex, and we're targeting incurable cancers. So we all know somebody who's been affected by cancer. It is still the second leading cause of death worldwide, and that's on the rise as we're living longer as a population. It is costing us over $100 billion to treat cancer annually, but only half this money is going towards targeted therapeutics. So why can't we better target cancer? That's because current therapies do not go after the causes. We all know what causes cancer. It's accumulated mutations in our DNA that cause cells to proliferate too fast. And chemo is something that we've been using for the past 30 years, and these are agents that target the DNA itself, damaging it beyond repair, and hoping to kill cancer cells that way. But besides the side effects that go with chemo, is actually allowing cancers to evolve resistance against it, because some of the mutations that the cancer benefits with the chemo actually end up being beneficial to the tumor itself. What we need to do is we need to go after the actual mechanistic causes within the cell, the communication network. Each cell in our bodies is composed of an average of 650,000 different proteins that are interacting with each other in the so-called protein communication network. Within these massive pathways, there are specific sub-pathways that are responsible for functions. There are pathways that tell the cell when it needs to destruct itself and die in a process called apoptosis, and there's pathways that signal for the immune system to recognize the cell and clear it from our bodies when it becomes mutated. Cancer bypasses these pathways. It ignores cues to destroy itself, and it ignores cues that tell the body to destroy it as well. However, by becoming that, it becomes actually really dependent on other pathways, such as the DNA repair pathway, that it has to upregulate to cope with the large amount of damage that is caused by replicating too fast. What we do at Syntex is we identify networks that cancer cells rely on, and within them we identify specific nodes that are now essential for the entire network to function, and we block them. This selectively kills cancer cells without affecting normal body cells in a process called synthetic lethality, ergo Syntex. So traditionally, in the drug discovery process, to get to a point where you have a compound, you know how it works, and you see efficacy in tissues, it takes $10 to $15 million and up to 6 years. This is a really long process, and we aim to disrupt it. At Syntex, we've developed our first compounds, STX-101 and 105, and we have great efficacy in vivo, and we've gone from concept to compound in 2 months with $20,000. So how do we do this? We have a proprietary drug selection technology that we've designed and developed. We have a cell that's reconstituting the interaction that we want to block, and in the same cell, it's making 1 out of 10 billion different compounds that can block it potentially. And these prodrugs are being selected by the cell. So we engineered it so whenever there is a constant interaction, it triggers a suicide cascade, and the cell destructs itself and drops out of the population. However, in the few instances where the actual compound breaks the interaction, the cell can now survive, and we can detect what caused it. We call this technology Torpedo. The platform starts with identifying the crucial interaction, screening an unbiased library of 10 billion different compounds, zooming in on the ones that work, and reiterating the library based on those to make it even better. And after we have the initial compounds, we test them in the clinically relevant models, such as patient biopsy samples. It is much better than the existing competition for these types of compounds, as they are made by the cell. They are so cheap because the cell is making them for us. The cell is also trying to survive, so they're reiterating the compounds themselves to make them even better at disrupting. And having it be intracellular also rules out anything that's generally toxic to the cells. It is cheap and fast. Syntex owns all the IP for the platform, the compounds, as well as the novel interfaces that we're going after, and we filed provisional patents to protect this with the help of Wilson Sonsini. So the lead compounds, STX-101 and 105 that I previously mentioned, block a really essential DNA repair pathway. This pathway is upregulated in a variety of cancers, and they depend upon it for survival. Some of the cancers are shown here: castration-resistant prostate cancer, lung cancer, pancreatic, and liver cancer. These are large patient burdens, but also huge market opportunities. We started with liver cancer. Cholangiocarcinoma is the second most common type of liver cancer, and it is on the rise as well because of bowel IBS and inflammation. It is very deadly. The current prognosis is 8 months with the standard of care. That's abysmal. But starting with the smallest cancer allows us to get an orphan designation by the FDA, which reduces the cost of clinical trials by half, allowing us to go into the clinic on our own. So here I'm showing you our results. This is a movie showing normal normal cells on top, the liver cancer in the bottom. On the left, they're untreated, and on the right, they're treated with our compound STX-101. The compound itself is labeled in green to show us that it is entering cells efficiently. And in the media of these cells, there's a dye that turns them red whenever they're killed. So as soon as we start imaging, we see that there's quite a bit of cell death, and it is very selective to the liver cancer cells. Normal cells are green, they're uptaking the molecule, but they're completely fine, they're unaffected. This is amazing because it's showing that we get selectivity on the molecular level based on a hypothesis that we generated by what the cancer is actually doing. And this is after 3 days. As you can see on the left, the liver cancer took over the complete population, but with our compound, there's no growth and no resistance. Furthermore, this happens really fast. This is as soon as we started imaging, and within 15 minutes, it was a complete death field. So this is very exciting to us. The kinetics are so remarkable. We asked around with different experts, and nobody's seen anything work this fast. So this is a novel pathway that our interaction is involved in, the control cell death. This is really exciting to us. So the next steps right now, once we have the compounds, we know what they're doing, we know that they work even in serum, we're putting them into animals. We'll be doing mouse trials in the next few months, and we're very excited to see that data. We have a pipeline full with different indications for the other cancers that I mentioned previously, as well as for things that I haven't had time to mention, as well as different interactions that we're going after with STX-201 and 301. Torpedo is going to be used in-house, but it's open to partnership with different pharmaceutical companies to develop compounds for them. We're furthering STX-101 to a point of an IND, and then at which point we will license it out and use the revenue to create more compounds that we will further in-house. We have a lot of interest from pharmaceutical companies in the platform as well as in the compounds, and we're currently in talks with several of these. We're only starting with oncology. We're going to use Torpedo as our engine to build a large, sustainable therapeutics company that betters human health by reaching all sorts of aspects of it. The founding team is composed of myself and Charlie, with over 25 years of research experience, specializing in cancer genetics and drug design. And we're advised by Tri Long, our business advisor, and we're very excited to have Leonard Post on board as our clinical development advisor. He's the CSO of BioMarin, and he's got a lot of experience bringing drugs to market. We're only starting with one interaction right now, but there is 650,000 more to go in our cells where we can modulate their individual functions. Help us make it a reality. We're currently raising $3 million to get us through our preclinical work and into an IND stage. We have $1 million committed. Thank you so much.
Hello, everybody. My name is Alec Lee, and I'm the CEO of Ava Labs. The future food is not organic, it's synthetic. And the proof of that will be in the sustainability, the scalability, and the ethical advantage of these methods. We're producing synthetic wine without grapes or fermentation. In 1976, this wine, a California Chardonnay, changed the face of the wine industry forever. In a blind taste test in France, against French wines, with French judges, this wine beat out all of the others and forever solidified the status of New World wines. Today, only a handful of these bottles remain, and if you can find one, they fetch well over $10,000 a piece. Soon they'll be lost to time forever. We can recreate bottles like this, or even digitally perfect our own, for a fraction of the cost of the originals. All food, and indeed all wine, is nothing but a collection of molecules. By analyzing the molecular profile of wines, we can rebuild those originals molecule by molecule. But of all the things to create synthetically, why wine? There are two core reasons why we focus on wine. The first is our technical abilities. Wine, compared to many other foods, has very low molecular and texture complexity. And that means we can rapidly bring to market products that match and even surpass the quality of their natural counterparts. The second is the market opportunity. Because of our technical feasibility, we can create wines that are dramatically lower cost than traditional wines, and people are willing to pay a premium for wine. And so those two drivers mean that we can generate high margins from day one. Now, the foundation of our technology is in our ability to digitize these flavor profiles. The wine industry has a long history of altering and adulterating the flavor of their wines, but this is the first time that we can bring to market a product, a wine, that has been engineered completely from scratch. And we've made a lot of progress along the way. We've already identified the key molecular ingredients for our wines, and we developed a prototype that people enjoy as wine. We control every molecule that's in our wine, which means we can have a product much higher quality and consistency than any natural fermentation process. Our ingredients are sourced pure from highly scalable sources, which means we can dramatically reduce the risk of crop disease, climate change, contamination. And unlike traditional wines, we can produce an unlimited amount of hours year-round, iterate rapidly, and match market demand in a way never before possible with wine. Now, the establishment is skeptical, but we've already submitted our wines to blind wine competitions. And while I don't want to reveal who they are because they don't know we did it, all 6 of the judges were fooled into believing that our wine was real, and they all rated it as good. I have the official results over at our booth later. The establishment is skeptical because there's a big nest egg to protect here. There's a $40 billion a year market just in the US, and it nearly doubled in the last 10 years. Now, nearly 70% of our pre-orders are coming from millennials, and that's where we believe our core target is going to be. Millennials decreasingly buy based off of the history of the vineyard, terroir, and increasingly buy based off price, taste reviews, the flashiness of labels, making them a perfect audience for a product like this. And millennials are expected to increase their share of the market threefold over the next 20 years. And we can do this all relatively quickly. The FDA provides minimal guidance on alcohol, and we're already in discussions with the TTB, which does regulate alcohol production. We have the freedom to operate and replicate the molecular profiles of existing wines on the market, and we already have provisional patents filed on both our process and our product. This is my team. Both Martin and myself have backgrounds in biotechnology and have founded multiple companies between us. Josh is our wine guru, also having a science background, and was one of the youngest ever to be certified sommelier in Canada. I'm happy to announce that we've raised a $2.5 million seed round, and I invite you to pre-order at avawinery.com. We're looking to make partnerships with distributors and retailers, and I'd also like to invite you over to our booth later for a taste test. Thank you.
Hello everyone, I'm Charvi from Knox, and we believe that asthma attacks can be prevented. This is Kai. When he was 9, he was walking back home from school, and suddenly he started wheezing. He could no longer breathe and felt as though something had been stabbing at his chest. The next thing he remembered was waking up at the ER. Kai's parents were even more scared. By the time they had seen any symptoms, it was too late. Asthma affects 40 million people, with 10.3 million children being affected. Kids are half the hospitalizations. But what if Kai's mom caught a problem 5 days before symptoms even escalated? The technology to do this exists. It's called spirometry, but it's trapped in the hospital because it costs $30,000 and requires a lab tech coaching you. So it only exists in 25% of hospitals. And even if you get this test, it happens every couple months. Spirometry is important because it captures lung obstruction, which occurs way before you have symptoms like coughing and wheezing. And this is when action should be taken. Therefore, we're developing Amaryllis, a portable spirometer that has hospital accuracy while being fun and easy to use. Kai might feel fine, even when he's not, but by the time he has symptoms like wheezing, it's too late and he's hospitalized. Amaryllis empowers families to catch the problem early on, so then they can immediately start the recovery process. We just completed an 80-subject study at UCSF last week, and as you can see, the Amaryllis results are the same as a hospital spirometer. Powered by Bluetooth, kids test their lung health by playing a game, and this is how they test their lung health. So your breath sends a hot air balloon into the sky as you travel to a new destination every day, and then you hear a story about that place. This is how we ensure that spirometry is done correctly, and it's also how we keep kids engaged. Today, Kai might feel fine, but lands in the yellow zone. Our app alerts his mom to start taking medication right away, so then he can get back into the green. We've placed Amaryllis into the hands of 8 families at home, and they loved it. Here's what Julianne's mom had to say.
value about Eris is it would help with the uncertainty. That's one of the highest costs of having a kid with a chronic health issue is you just never know when things are going to fall apart. So if you could get that early warning, it would really help because you could treat it and cut it off at the pass, but you could also plan better.
In the 6 weeks we've opened signups, we've gained 4,000 subscribers who are waiting to be notified when Eris is available for purchase. Last month, we received the prestigious $250,000 National Science Foundation Award for our work in developing a machine learning algorithm that ensures that our test is as consistent at the hospital, but we can do it without a lab tech. So Eris will be available for purchase in spring 2017 on our website, Apple, and Amazon for $180 at a 50% margin at retail. A $10 per month subscription provides you with news story content and alerts of how environmental factors affect your health. And unlike our competitors, our deep learning algorithm ensures that only properly collected spirometry measurements are accepted and then acted upon. We're also hyper-focused on children ages 5 and up to create an experience that they love using, day after day for years to come. My background is in bioengineering from Berkeley and UCSF, and I was previously a software engineer at Genentech developing algorithms to determine medication efficiency. Hyson leads software, and it has expertise in mobile and cloud development. Indy leads hardware and previously worked at Equifax developing enterprise-level software. And Yufei leads mechanical engineering as a former Apple product design engineer. We're working with 2 UCSF pediatric pulmonologists, and our technical advisor is Jim Rosen, who served as CTO for 3 companies with 2 exits. Our mentors include Alan May, the chairman of the Life Science Angels, Patrick Scoglia, the former CTO of HP, and Chriso, the founder and designer at Starters. Since IndieBio, we completed a validation study at UCSF. We've had 8 families try Eris at home for a week and received a $250,000 NSF award. This month, we'll be submitting our 510(k) application, and next month, we'll be launching our ResearchGit app for a 500+ subject NSF study. We anticipate FDA clearance in December of this year, in time for product launch in spring 2017. And once we gain a strong foothold within the pediatric market, we'll then expand to the larger adult population and other respiratory diseases like COPD, cystic, and pulmonary fibrosis. We have $100K committed so far, and we're seeking to raise $1 million for an additional hire, go through FDA clearance, and also begin pilot production of 200 units. At Knox, our goal is to convert knowledge to action so that you're in control. Thank you.
Hello, my name's Jeremy Warren, co-founder and CEO of Astron Biotechnologies, and we're putting food safety in the palm of your hand. This is fear. This is the fear every company has, that they're going to be known for one thing, and that's a headline. 1 in 6 Americans get sick every single year from foodborne illnesses. And when people get sick from the food they eat, they lose trust in the companies providing their food. And this is what a loss in trust looks like. Chipotle lost over $8 billion in market cap in 1 quarter due to 2 very highly publicized food safety outbreaks. And guess what? They haven't regained the trust of the customers, and they haven't regained their valuation. But Chipotle is not alone in this. Produce companies are especially susceptible to outbreaks due to salmonella, listeria, and E. coli. But the companies realize this. They spend over $4.5 billion every year to ensure that your food is safe. But if we're spending so much money, why are people still dying? The reason is current testing methods take too long. And your food can be contaminated at any step in the supply chain, from the farm, through the facility, and all the way to your table at home. But why is it taking so long? The current testing methods are broken. It takes way too long to get your results. Currently, companies take a sample, box it up, send it to a third-party testing lab. At the lab, they then have to grow and culture the bacteria. This takes another 24 hours. This means companies can wait up to 5 days before they get their food safety testing results. You can't possibly take 5 days at every step in the supply chain. Product lifetimes are finite. This leads to companies sending product before they know the results of their food safety testing. This leads to recalls. This leads to people getting sick. This leads to people dying. And the recall itself is very complicated. They have to go back to the facility, track the lot numbers, go out all the trucks to the stores where the food was sold. This whole time, people are still getting sick. But what if there was a better way? What if you could test at every step of the supply chain and know that your product is safe before you ship it? We at Astron Biotechnologies have that solution. Using our proprietary RNA-based detection platform, we are able to detect pathogen presence at the point of need in 1 hour. We do this with 4 easy steps: capture, wash, amplify, and detect. We're going to talk a little about science here, but I'm going to go through it quickly for you guys, all right? Our product fits exactly into what companies are already doing. Simply take your swab of the sample you want tested. You're going to put that into our first buffer. This physically breaks open the bacteria, releasing the RNA. We choose to detect RNA because, unlike DNA, which has a single copy, there could be tens to hundreds of thousands of RNA copies per cell. And if we use this in conjunction with our proprietary capture protein and sequence-specific tag, we can eliminate culturing altogether, providing results at the point of need. Then we just isolate the pathogenic RNA we want, wash away everything we don't. Then we developed our proprietary isothermal amplification to create a signal from that RNA and amplify it. Then it's as simple as detection. If the solution turns red, it's contaminated. But this is the science. How to make this into a product. This is our product. This is the fully disposable Astron Biotechnologies disposable kit. Each kit can detect one pathogen, say E. coli, Salmonella, or Listeria. We have sensitivities with the USDA guidelines, and we have in the lab, we have done 10 cells, and in the field, this kit has gone out in the field with our pilot programs, and we can test 100 cells using this kit. We have 4 companies signed up for our pilot program. We have already been to 2 of those companies. We have shown them how easy food safety testing can be, and they have reiterated the need for fast, easy results. Our pilots are mainly with small and medium businesses. This will be our initial market. Targeting these businesses will unlock a $200 million market opportunity. But this is only the first step. The next step in the vision of Astrona is the fully automated machine. One single user input, put your swab into the cartridge, and put it into the machine. The machine does the rest. It tells you which bacteria are there, how much is there, and it'll automatically record the data for you. The reason we're making this machine is because the large corporate businesses told us they wanted it. By accessing these businesses, we open a market opportunity of over $2 billion. Here's the model. These kits sold to small and medium businesses for $50 a piece. They cost us $5 to produce. These companies are buying between 100 and 1,000 kits per year. The machine works in a very similar way. We're selling test cartridges, $50 a piece. These will be around $5 to produce as well. We roll the machine cost into the initial purchase price of the test kits, and they can have the automated data tracking software if they want it. These are our company milestones. These include third-party certification, initial test kit rollout, and prototype development of the machine. Of course, there are competitors in the landscape. These mostly consist of third-party testing lines, which take 3 to 5 days for results. There are other companies that are getting in within 1 shift, but nobody can detect bacteria, viruses, and fungi at the point of need in 1 hour like Astron Biotechnologies. Here's our team. I have over 15 years experience in molecular-based solutions. Peter Ort, Mark Pollock, our CSO, started making this technology at UC Davis, and our first hire is Peter Ort. He has over 15 years of experience in startups. And we also just hired an intern, Joe Congerti, to help us with marketing and business development. We have a great advisory board of scientists from the #1 agricultural school in the world, UC Davis, consisting of RNA biochemists, food safety specialists, and engineers. We also have a business advisor, Mike Bowles, to help us keep our business on track. We're looking to raise $1.7 million for general kit rollout, prototype of the machine, and initial hirings. Please meet us at our booth if you want to change the future of food safety. Thank you very much.
Hi, everyone. I'm on. Yep. My name's Shane Greenup. I'm the CEO of Nascent. At Nascent, we are building better health outcomes for babies who aren't breastfed. Milk is nature's perfect food. It is the only food which is highly evolved and precisely designed to give baby mammals everything that they need. Feeding babies anything other than mother's milk means they won't get the best nutrition for growth and development. This fact has been shown in many studies. Babies who are fed anything other than breast milk risk suffering from a range of health and lifestyle disadvantages. Higher rates of obesity, diabetes, infection. They suffer from higher overall mortality. These facts became very personal for me soon after my son was born in 2014. My wife suffers from a serious chronic illness, which meant that she struggled to continue breastfeeding. And so we had to start supplementing my son's diet with infant formula when he was just a few weeks old. Now, our situation is not that unusual. Fewer than 19% of US mothers exclusively breastfeed for the recommended 6 months. So a lot of families are relying on infant formula. So what is infant formula? Originally, it was just the milk of other animals. Cow's milk was, of course, the most common, but the babies that were fed it tended to suffer from scurvy and rickets, so we added orange juice, which is vitamin C, and cod liver oil. They still weren't thriving, so more vitamins and minerals were added. Malted wheat, honey, vegetable oils, corn syrup solids. The whey protein concentration was increased. DHA and ARA from fungi and algae. Most recently, customized oligosaccharides have been added. So you can see infant formula is an incredibly complex food with a long history of improvement. And yet still we see these negative risks to the health of infants. So the problem here is that for all of these improvements, what we still have is ultimately cow's milk. And milk is a species-specific food. So baby humans need human milk, and that's what we are doing. What I have here is human alpha-lactalbumin. We put the gene for this into Pichia pastoris, which is a strain of yeast. And that yeast we used like a microfactory to produce this natural human breast milk protein. This technique is fundamentally no different to what has been used for the past 30 years to make human insulin for diabetics. This is the first of many proteins. It is the first because it is the key nutritious protein in breast milk, taking up 35% of the protein of colostrum. It is rich in essential amino acids, and easily digested by babies. This formula here, protected by a provisional patent application, is the world's first human formula, a prototype of the world's first human formula for babies. As each of these proteins are produced, they will be added to this bottle, and it will contain the next best thing to human breast milk. Our next step is to work with the FDA to have our alpha-lactalbumin granted the generally recognized as safe status. As soon as that happens, we'll be the creators of the world's first recombinant human protein food additive. We've already begun working with an expert GRAS consultant who has a 100% success rate over their 10-year history. And our conversations with the FDA indicate that they're incredibly excited by the work that we're doing and eager to help us as much as they can. As soon as our protein is granted GRAS, we will begin with products that help the most vulnerable babies. In NICUs, they have to add additional nutrition to milk to help premature babies catch up with the normal birth weight. Right now, there's only one company selling nutritional fortification from human milk. Prolacta buys milk from women and makes a fortifier out of it. Unfortunately, there's always a massive shortage of donor milk. The demand is 5 times the supply. So this approach is simply not scalable. It also makes it incredibly expensive, at $10,000 per baby. We will be able to produce human protein for milk fortification much cheaper than that within a year. This is what that looks like. We can sell our protein right now as a research reagent at a significant profit. We have partnered with Biomass, and they are selling our protein for us, and we can use that to create a revenue to get us through optimizing, scaling, and testing of our protein. As soon as we reach the $50 per gram price point, we can start working with NICUs and using our protein as a human milk fortifier. And that will help feed the 400,000 premature babies born every year. We'll continue to scale and drive down our price until we are able to meet the needs of the infant formula market, at which point we will start selling our own infant formula and providing our protein as an ingredient to other formula companies. We have a letter of intent from Invita, which is Brazil's fastest growing infant formula company, to purchase our protein. And we're in private talks with one of the world's largest brand names. This will lead us to our ultimate goal of replacing the entire infant formula, $20 billion industry. In the future, when you go into supermarkets, you won't be choosing between cow, goat, and soy formulas. You'll be choosing between specific formulations of human protein mixtures, and we will be at the center of all of it. The team bringing this into reality is an amazing set of complementary skills. I've been building businesses for 10 years after graduating with honors in molecular biology. Craig Rowski has 15 years of experience in molecular biology and immunology, and Terry Anderson is a communications strategy expert and serial entrepreneur. We have an amazing team of advisors guiding us through this process, from pediatrics to infant nutrition and business development. We're raising $2 million in this seed round to help get us through the next 18 months of optimizing, scaling, testing, and approving of our proteins. We invite you to join us on this exciting journey and help us build better health outcomes for babies who aren't breastfed. Thank you.
Hi, I'm Brad, co-founder and CEO of Amaryllis Nucleics. And at Amaryllis, our mission is to improve RNA sequencing to accelerate discoveries in life sciences and improve human health. But why do we want to sequence RNA? Well, most people are familiar with DNA, but DNA is static, and every cell in your body has the same DNA, so it doesn't tell you what's happening. RNA, on the other hand, is dynamic. Every type of cell has a unique fingerprint of RNA, more unique than a fingerprint. But it doesn't just tell you what a cell is, it tells you what a cell is doing, what it's responding to in its environment, and how it's diseased. And the implications for us are that RNA can be used to identify illnesses even before the first onset of symptoms. It can even, on occasion, be used to determine which chemotherapies a tumor will be responsive to before having to give them to a patient. And eventually, in the near future, RNA-based tests will be a routine part of every doctor's visit, and we want our technology powering those tests. A future in personalized genomic medicine and stem cell therapies require easy and efficient access to RNA sequencing, and that's what we provide. The importance of RNA sequencing is reflected by the rapid adoption of this technology by the scientific community. The number of journal articles using RNA sequencing are increasing exponentially. And this importance is also reflected by the size of the market. $447 million in kits for preparing samples for RNA sequencing were sold last year. And this is a 17% increase over the year before. And with increased adoption of this technology in diagnostic medicine, we see this growing even faster. But a major roadblock to full adoption of RNA sequencing is the cost and complexity of preparing samples for RNA-seq. In order to sequence RNA, you have to do a complex series of chemical reformatting so that they can be read by machine. And this can consume up to 80% of the cost of an experiment, forcing researchers to have to cut back to a bare minimum or not do an experiment at all. So when we were scientists at UC Davis, we had to prepare thousands of samples for RNA sequencing. And in order to do our work, we had to find a better way. So to do this, we took advantage of a phenomenon called terminal breathing. And what that is when DNA, RNA, and cDNA in their double-stranded forms open and close at their end. And when they open, this is when our adapter can attach without ligation, streamlining the process. And just like Taq polymerase made PCR a routine procedure, using breathing to prepare the libraries for sequencing makes the process incredibly efficient. Competing technologies can take as long as 2 days and have many manual steps, increasing the likelihood of failure and error. With our technology, we can complete it in just 4 steps in as few as 4 hours. And when we compare our data to industry-leading Illumina's ScriptSeq, we're 17 times more strand-specific, dramatically reducing errors. And we can do this all at a much lower cost. Also, RNA sequencing is notoriously bad at capturing the beginning of RNA molecules. And these regions, called UTRs, are critically important for regulation of cell function and oncogenesis. But with our technology, we can capture these regions effectively, which improves biomarker discovery and diagnostic testing. So to get our technology into the hands of the people who need it, we've created this fully self-contained kit, which researchers can use in preparing their own samples. And we're selling it to academic researchers and sequencing facilities. But in selling these kits, we found that many researchers wanted the reassurance of having other experts handle the experiment for them. So to that end, we created an end-to-end service where scientists send us their samples, we handle the sample handling, sequencing, and return them analyzed data. And this provides us with 2 revenue streams. And our business model works. In 4 months, we've $230,000 in sales and work orders, and $420,000 in quotes we expect to close. Thank you. And we're getting traction, not just with revenue, but with the kinds of partners and customers we're hoping for. Transformative institutions like UC Davis, UC San Francisco, and the CDC. And these are all heavy users of RNA sequencing and key influencers. And we've chosen them because others will follow their lead. The competitive landscape for RNA sequencing puts us in competition with giants like Illumina, but we can beat them on speed, on cost, and on quality of the data. And this positions us to take a large share of the market. We have exclusive IP on our technology, and we've published it in a peer-reviewed journal. We called it BradSeq, and my name is Brad, but I'm not the one that named it. And we've been using this in our own work for over 2 years, and it doesn't just work, it works beautifully. So we're growing fast, and right now we're working on automation to scale our services. And we can leverage this automation to make robots used in high-throughput sequencing facilities work with our kits. And we're talking with reagent manufacturers to outsource our production and drop shipping, and soon we'll be in non-US markets. Our founding team is composed of UC Davis scientists with deep backgrounds in molecular biology and bioinformatics, and together we have over 36 years of molecular biology experience. So to fast-track our growth, we're looking to raise $1.5 million, but we're also looking for partners and customers. So if you're doing RNA sequencing, please come talk to us. Thank you.
Hi, everyone. My name is Alan. I am the CEO of Miraculics, and we are creating the future of sweet. Because just like Homer, I'm addicted to sweet foods. And unfortunately, we all share that same gene to crave the sweetness. But as well, we're sort of learning what this gene is sort of resulting in. Heart disease, diabetes, metabolic syndrome. All of these things are occurring because we crave the sugar, and it's everywhere. Governments around the world are realizing this, and that's why they're putting in new regulations that were never seen before on sugar. New labels, new taxes, new warnings on everything. The sugar industry and the sweetener industry is worth over $87 billion, and it's growing 5% every single year. It is full of sweetening products that hurt your health or don't give you the taste that you want. So what we are doing at Miraculics is we are creating the first generation of great tasting sweeteners that are actually healthy for you. These are protein sweeteners. They come from rare exotic plants, and we have a technology to mass produce these proteins that they produce. We are starting with Miraculin, named for its amazing ability to convert sour into sweet. It has 0 calories, and it provides 30 minutes of amazing taste. This is truly a miracle. So don't only take my word for it. Take the word of these people who even say it tastes better than sugar.
Wow.
Easy to it. Really easy to eat it. Wow. Jesus Christ. Wow. Are you serious? Yeah, it's like candy. That was really cool.
Wow is the word. Protein sweeteners can make that wow. So what protein sweeteners are large proteins that are much bigger than traditional small molecule sweeteners. And what they do is they could bind to your sweet taste buds like no other sweetener can. In fact, there is a lot of scientific evidence showing that many of these protein sweeteners actually evolved alongside humanity. So in essence, they taste amazing, 0 calories. And what do? They fall apart into amino acids after you eat them. And not only that, a tiny amount of these protein sweeteners are equivalent to over 20,000 grams of sugar. So people have asked me, why hasn't this been done before? Why isn't this stuff everywhere if it's so amazing? Unfortunately, it's really hard to produce. In fact, there are very few places in the world where you could actually grow it. It requires a specific climate, temperature, soil, and different care. So even with biotech, unfortunately, people couldn't really get the folding correct. The glycosylation wasn't correct. And even the cost of purification was still too much. So that's where we come in. We have a technology to hydroponically produce these amazing plants and the proteins which they come from. Today, we have managed to purify and concentrate these amazing proteins, and over here, we are producing it today. We have a priority system that we utilize hydroponics to grow these protein sweeteners like never before. We are producing it in highly concentrated forms, and we are looking to scale using biotechnology and recombinant plant systems that also use hydroponics to grow. The reason why hydroponics, it's a very well-known technology that's able to scale very quickly and inexpensively. And in essence, we could grow a huge amount of it in a very, very small footprint incredibly quickly. Right now, we have a projected cost of 32 cents per serving, and using our technology to scale, we aim to have about less than 2 cents a serving in a couple of years from now. We are talking with incredibly large food producers who are very interested in bringing better tasting sweeteners to the market. Our production timeline is to bring Miraculin to the market by this year and bring in other protein sweeteners that could be replacements for sugar within the next 2 years. Our projected milestones are to expand our hydroponic systems, bring in new sweeteners, and to bring these proteins to everyone. We currently have 2 patents pending that would protect us as we move forward to bring our products to market. Our team is made up of plant biologists, protein engineers, organic chemists, and food scientists who know how to bring these amazing products to the world. We are looking to raise $2 million for strategic partnering, expanding our portfolio, and for key hires. The reason why we're doing this is we could finally allow everyone to have their cake and eat it too. if you would like to taste these amazing protein sweeteners, come visit our booth at the end. Thank you.
Everyone ready to look great? Hi, everyone. My name is Carolina. I'm the CEO at OneSkin, and we are creating this unique skin-based platform for the next generation of cosmetics. Why should I care about my skin? Why should you care about yours? Our skin is the largest organ of our body. It plays such an important role for our health. Through our skin, we interact with the external world. We feel the wind, the sun, the pleasure, the pain. Also, our skin is our identity, and that's why we care so much about it. This makes the personal care one of the biggest industries in the world, and only for anti-age, it is expected to reach $36 billion by 2020. But when you go to the store, how do you know if that product works for you? Well, we actually don't know. We are just buying blind. And we end up spending tons of money looking for the right products. And here are the reasons why products are not effective. Companies are still using out-of-date technology to develop their products. As we all know, we are not likely a dog. We are not represented by a single layer of skin cells. And most people don't fit in that standard Caucasian skin model that many companies are using. This brings the needs for models that represent our diverse ethnicities and also different ages. And I see here a great opportunity to apply the science that have been developed for many years. And with our team, we are bringing a revolutionary solution. At OneSkin, we are growing real human skin. And we are bringing our beautiful diversity at one platform. So now you are looking to the skin that we grow in the lab. And it's formed by the most important layers of the skin, the derms that provide the firmness of the skin, and the epidermis that forms our main physical and immune defense barrier. And now you can see how similar is the skin that we grow from natural human skin, matching them layer by layer. Now I'm going to explain how we build our platform. So we first grow the human tissue, and in order to do that, we isolate cells from different donors to represent different ethnicities, and we then reconstruct the human skin tissue in the lab. In order to produce skin at different ages, we submit these skin models to UV, and then we accelerate the aging process. And then we can test products in skin that now are 20, 40, or 60 years old. But how can we measure the age of these skin models? And here is where we bring our innovation. We are going to isolate the DNA of these models, and we run this DNA data to an algorithm that's called the molecular clock. This algorithm correlates the DNA alterations with the age, and we are the first and the only company to apply this technology for skin care development, and we already filed our provisional patent. So here you can see an application of this algorithm. We have a skin model at a certain age, and once we want to test a product, if that product works for rejuvenating or for accelerating the aging process, you can see it by the spots in this chart. And why is that important? Because now we can guarantee consumers if the products that they are using for anti-age are really effective or not. Our DNA doesn't lie. Here you can see we're running our platform. So we are growing hundreds of human skins. We then submit them to a solar simulator to induce this in vitro aging. And finally, we are testing this anti-aging product. And we are already anticipating the need for personalized skincare products. And that's why we are creating skins that represent different ethnicities, ages, gender, and we are the only company that is using this molecular clock to validate the efficacy of anti-aging products. Our competitors are still producing standard models or only one type of aged model. And we already got early tractions, and one of our customers is this international skincare company, Nerion, and they want to have their products validated by our platform with an initial demanding service of $150K. And we are also happy about this partnership with ST&T. They run clinical trials for several cosmetic companies, and this will work as a great channel for bringing new customers. We are starting generating revenue, providing this service for companies as Nerion and other anti-aging companies, but our ultimate goal is to develop our own product, a science-based skincare line. And at this point, we will achieve an exponential growth, selling our product through e-commerce and partnership. It's impressive what we have achieved in less than 4 months, but we are more excited for what is coming now. Soon, we'll be producing the ethnically diverse skin models, and in less than 2 years, we expect to launch our first skincare line. And I can already see the success that this is going to be based on the early results that we are getting now. The amazing team that is bringing this dream to a reality is formed by these 3 powerful women. We gathered a very unique expertise together, combining backgrounds in tissue engineering, stem cells, and bioinformatics. And we are also supported by these exceptional advisors. Sylvia has more than 20 years of experience in skin biology, and Ricardo has helped us a lot with his experience in business development and fundraising. We are now raising $1.5 million. That's going to accelerate the process of launching our first product, and we are looking for partners in the cosmetic industry. And also, if you want to help us to build this personalized skincare line, come talk to us. Thank you.
Hi, everyone. My name is Sarah Bonham. I'm the CEO and co-founder of Willow Cup. We craft premium plant-based dairy indulgences to power a food system that will sustainably take us through the next 100 years. We're in a dairy revolution. Never before have we seen such an unprecedented shift in what consumers are asking for in the $440 billion dairy industry. You see, cow's dairy has been on the decline in terms of consumption per capita for the past 25 years. This is driven by environmental concerns, health concerns, and shifting demographics. 65% of the world is now lactose intolerant. But behind these challenges lies an incredible opportunity in the alternative space. Specifically, with plant-based dairy, this market's gonna explode, go from 10 to 19.5 billion in just 4 years, showing no signs of slowing down. And it's predominantly driven by on-the-go, health-conscious millennials who represent $1 trillion buying power. Never before have we seen a generation care so much about health, wellness, experiences. And at Willow Cup and our protein technology platform, we're gonna own this space. I know what you're thinking. What about all these other plant-based dairy alternatives out there? Let's address that. While they do have their advantages, their functionality is their fatal flaw. I mean, you can't really add soy milk to a coffee that's more on the acidic side without it curdling because its proteins are so sensitive and highly charged. To compensate, we add all sorts of emulsifiers, stabilizers, unhealthy flavors, and This is in no way indulgent and not acceptable. So, to truly build a revolutionary plant-based dairy product, we took inspiration from the original, milk. We looked at cow's dairy under the microscope to really understand what gives it its luxurious properties. And it's these multiple proteins, like casein and whey, that work together to give dairy incredible versatility, functionality, and complexity. We took this approach when developing our core dairy technology. We asked ourselves, what if we took the best protein profiles, processed them under advanced techniques, tuned their mechanical properties, and rebuilt a better plant-based dairy, molecule by molecule, one that had functionality, that didn't curdle when you added it to a coffee, one that had creaminess, richness, and the sensory properties of the dairy you've grown to love. And we did it. This is our dairy under the microscope. And you can see from the middle graph that it's on par with cow's dairy in terms of its structure. We have an advanced process that allows us to break apart healthy fats and adhere them to the surface of plant proteins. From there, we can rebuild a better protein matrix that's on par with cow's dairy, one that's homogeneous, and this in turn relates to better properties, functionality, sensory, rich, creamy taste, and the luxury indulgent products that you've grown to love. Not the grainy, chalky taste that's often associated with a lot of the plant-based dairy products on the market. Our rational approach and advanced protein technology platform has allowed us to create a plant-based dairy like no other. This is our Willow milk. It's the heart of our technology, and we're going to use it to power an entire line of high-margin, high-value indulgent products that are more personal than a glass of milk, but can make just as big of an impact in the world. And I'm excited today and proud to introduce our first product, the Willow Milk Whipped Latte, right here. Oops. This is like nothing you've ever tasted. It's creamy, it's rich, and when you shake it, it actually foams like a real latte. We've designed this product molecule by molecule with our proteins so that you can have your favorite latte anywhere at any time while sailing on the Pacific or hiking up Twin Peaks. The ready-to-drink market is also the largest of the indulgent category, growing at an astronomical rate and ripe for cleaner label, healthier products. Our go-to-market strategy is simple. We've received incredible traction since our time here at IndieBio. By the end of the year, we're going to be in 12 locations in the Bay Area, and from there, we're going to leverage e-commerce distribution to cater to the on-the-go, health-conscious millennials. While we scale up our operations and distribution, we're going to find a co-packer, and within 1 year, expect us to be all over California in Whole Foods, Costco, you name it. We're already profitable. It only costs us $1 to make our formulation. We sell it at $4. And with time and volume, we can bring this down to just cents. Yes, there are many plant-based dairy companies on the market. We consider ourselves a second-generation company for plants, inspired by our predecessors. What makes us different is our patented Willow milk. And we're going to use this to enter the market not as a commodity, low-margin milk, but a high-value, premium luxury indulgence. And yes, there are many ready-to-drink beverages, we know, but I ask you, how many of them are made entirely from plants? Not so many. And how many of them have no additives, stabilizers, emulsifiers, all this junk? And how many of them actually foam like your latte should? Willow. 150 million Americans had a cup of coffee this morning. 65% of those beverages were specialty products like lattes, and we're going to sell millions. We've already started. Since our time in IndieBio, we've powered over 250 beverages in the Bay Area. But we're not just a latte company. We're going to use our patented Willow milk technology to power an entire line of indulgent products to own this $40 billion category. And we've already begun prototyping. In our time here, we've developed ice cream, frosting, and that's just the beginning. I wouldn't be here today without my amazing co-founder, Craig Debank, and my team. We consider ourselves food explorers that are merging scientific discovery with design thinking to solve some of the world's greatest challenges. We have over 50 years' experience in bringing food products to the market. And I'm here today to ask for $1.2 million to bring on key hires, ramp up sales, and expand our manufacturing operations. If you're an investor or interested in partnership opportunities, we welcome you to come to our booth. As well, we have lots of samples. So stop by and taste the plant-based dairy revolution. Thank you so much.
Howdy, y'all. My name is Kyle Taylor, co-founder of Enduro Bio, where we're helping to protect crops from drought. All too often, it's easy to forget the farmers who grow the food that we eat. But these are the men and women who are the backbone of the $3 trillion global ag economy. And right here in our own backyard, California farmers bring in $35 billion in crop revenues annually. But there's a problem. Drought, fueled by climate change, is causing real economic damage to these farmers. UC Davis did a comprehensive study of the 2014 growing season and found that drought cost California farmers $1.25 billion. There simply wasn't enough water available, and the water that was available was more expensive. All this boils down to an urgent need for crops that maintain yield while using less water. So one solution to this problem is to do traditional breeding, where you take and select the most drought-tolerant plant in your field, cross them, grow them, and repeat that process. But this can take up to 5 or more years. Oops. Another solution is to engineer GMOs or use transgenic technology. However, there's still market acceptance problems. Consumers still don't trust GMOs. And there are higher regulatory burdens, all of which boils down to delays in getting these new varieties to the farmer where they're needed the most. And that's what these solutions miss. Farmers already have crops in the field that they need to mitigate against drought. So for example, farmers in California have a lot of fruit and nut trees. They're already in the ground. They can't go changing those up when a new variety comes in. So we can take insights from what farmers are already doing. Farmers are already managing crops in the field, such as applying fertilizers and pesticides. So what if we could apply something in the same way we do fertilizers and pesticides, but this confers some degree of drought tolerance instead? And that's our first product here at Enduro Bio. We're working on Climate Guard, a proprietary prepackaged mixture of enzymes and natural products from more drought-tolerant plants to confer drought tolerance onto crops already in the field. Farmers can use it just as they would normal fertilizers or pesticides. So how does it work? So we go after some of the key reasons for yield loss due to drought. Climate Guard has antioxidants to prevent oxidative damage, improves water use efficiency to correct water imbalances caused by drought, and primes the plant's stress response to ensure there's a minimal delay between when drought occurs and when the plant is adapted to that stress. This gives Climate Guard several key advantages. So it's fast, seeing results within around 2 to 4 weeks. We can customize it to vegetables, fruit, and nut trees that California farmers are already growing in the field today. And finally, we're compatible with the USDA organic label. So now I'd like to share with you a little bit of preliminary results. So we started off doing a pilot tomato study where we paired some tomatoes at the same developmental stage and same size. You can see representative pictures up here. We then treated the one on the right here with Climate Guard, and the one on the left was a mock untreated control. We then subjected them to water stress, simulating drought. And after 4 weeks, plants treated with Climate Guard were about 1.5 to 2 times bigger in size than the untreated controls. To further show the efficacy of Climate Guard, we then wanted to quantify the number of flowers each of these plants had, because you can't have a tomato without having a tomato flower. And our tomatoes treated with Climate Guard had a 15 to 25% increase in flowers, the bars on the red, in comparison to the untreated controls, the bars in blue on the left. So encouraged by these results, we went out to the Central Valley and started talking to farmers. We talked to around 12 or so farmers and ended up wanting to work with Steve Hash of Steve Hash Farms. He's a 20-plus year veteran growing stone fruit in the Central Valley and a former chairman of the California Fresh Fruit Growers Association. And as a professional plant bio nerd, I can say he has some gorgeous trees. So we worked closely with Steve to come up with a pilot experiment to test Climate Guard on his plums. And I'm pleased to say that we went down there just last week to start that pilot, and we'll have results of those experiments in the end of August, last week of August, first week of September. So what value do we actually add to farmers? So if we go back to that study that UC Davis did, if you dig into the numbers, farmers are paying, on average, around $200 an acre more for water. If instead they tentatively pay $75 an acre for 2 applications of Climate Guard, they end up having an increased revenue of $125 an acre. So that tentative $75 an acre breaks down for Enduro like this. So 35% is the cost of goods sold. 25% is traditional channel costs. This is important because this is how we're going to get Climate Guard to farmers directly using the same input channels they use to buy fertilizers and pesticides. Finally, that leaves a 35% gross margin for Enduro Bio. As I mentioned before, we'll have the yield results for our tomatoes at the end of August, and the yield result for our plum trials at the beginning of September. Over the winter, we'll then be optimizing the frequency of application, the stage at which to apply Climate Guard, and the method of applying Climate Guard, all of which is to gear up for field trials next summer, as well as getting organic certification. Finally, we hope to be shipping product the end of fall, the following fall or winter. We have a great team to execute on this. Shashank, my co-founder, is a serial entrepreneur, and I myself am a professional plant bio nerd with 10-plus years of experience. But more importantly, we both have a passion for agriculture and the farmers who grow our food. We're looking for any contacts you may have for farmers and growers who need help managing drought, as well as looking to raise $2 million in seed funding to get to the California farmers. We are Enduro Bio. We're protecting crops from drought. Thank you for your time, and I look forward to chatting with you after the pitches. Yeah.
Hello, everyone. I'm Pratish Kawant. I'm the CEO and co-founder of Ardra, and at Ardra, we make all-natural flavors and fragrances. We all were promised a bio-based revolution. Bio-based chemicals were going to revolutionize the chemical industry by making it more sustainable. But that revolution never happened. And that is because bio-based commodity chemicals could not compete with petroleum-based commodity chemicals at cost. There is not one single bio-based commodity chemicals company today that can make these chemicals at profit. But in a stark contrast, there are specialty markets waiting to be served by bio-based chemicals. Flavors and fragrances is a $25 billion industry with a clear consumer-driven preference for natural ingredients. But extracting natural ingredients from plants is expensive and inefficient. And this is where industrial biotech can help. By transferring natural pathways for making flavors and fragrances into industrial hosts, such as yeast, we can make these chemicals at industrial scale fermenters, at scale. And this is the current state of the art. but it is based on a fundamental assumption, an assumption that natural pathways are inherently efficient for making these chemicals and will work well at industrial scale. But let me tell you, this assumption is wrong. Natural pathways are inherently inefficient for making flavors and fragrances. Nature did not intend fruits and flowers to be factories for making these chemicals. And the current solution to this problem is to use brute force experimentation to optimize the natural pathways using high throughput robotics. But brute force experimentation does not always work. And given the advances in the field of industrial biotechnology and metabolic engineering, we don't have to limit ourselves to natural, inefficient pathways. We can design our own pathways. And that is what we are doing at Ardra. At Ardra, we design novel designer pathways that outcompete nature in making flavors and fragrances. This schematic here shows a core design on which every single Ardra's pathway is based. Our pathways are designed to use sugars or natural precursors. They are designed to give high yield, thereby reducing production cost. And most importantly, every pathway is designed so that it can be rapidly adapted to make multiple products. At Ardra, we are using one pathway to make many products. And this allows us to bring down the development cost and time of every new product we add to our portfolio. And we do this by using at least one promiscuous enzyme in every step of our metabolic pathway. Promiscuous enzymes are enzymes that can act on multiple substrates and can give multiple products, in contrast to more commonly found specific enzymes that act on a single substrate to give single product. And we have our IP around our designer pathways based on promiscuous enzymes. We filed a provisional patent while we were at IndieBio, and we have also licensed an exclusive license from the University of Toronto on a designer pathway that I co-invented. We came to IndieBio with a plan to answer the question, do Ardra's designer pathways work? And in the last 4 months, we have executed on this plan and found a definitive answer to that question. And the answer is yes. We started with making one product in a shake flask at the titer of 1 gram per liter. And now we have increased the titer of our product 10 times, making it ready for 300-liter pilot scale trial. But that's not all. We have also added 2 new fragrance compounds to our portfolio, thanks to our one pathway, many products technology. And we are not making these products in proof of concept titers. We are already making them in grams per liter titers. And this is the most important part. Our fragrance compounds become profitable at a titer of 10 grams per liter at a price point that is provided to us by our potential customers. And we have already demonstrated that we can reach this titer in a matter of months. And pictures don't do justice to our fragrance products. So our Chief Scientific Officer, Jonas Miller, went ahead and took a fermentation bath so that all of you here can smell Ardra's fragrance product. Please find taped to every chair here on the right side, a tiny vial that contains Ardra's fragrance products. I request you all to please go ahead and smell it. It's a tiny vial like this. Please do not taste it, just smell it. And what you are smelling, or will be smelling, is a chemical called leaf aldehyde, or trans-2-hexanol. And it smells like freshly cut grass, crushed leaves, or green apple. It's a green note and is used in flavors and fragrance industry to impart freshness to products. And leaf aldehyde is just one of our products. We have a whole portfolio of related products that give us access to a $500 million market. And our portfolio grows. Remember, flavors and fragrances is a $25 billion market. And over these months, we have received customer traction on every product we made, including our test product, 1,3-butanediol, which finds application in cosmetics industry. We have received unsolicited emails and phone calls asking for samples of our product. A large cosmetic company is interested in our fragrance portfolio, including the chemical that you just smelled. Givaudan, world's largest fragrance house, is interested in multiple products on our portfolio. And we can bring these products to market quickly. That's because we use a lean and nimble business model. We don't set up our own facility and keep the capital cost low. We just rent contract manufacturing facilities to make the products and sell them. We also partner with our end user customer, understand their product needs, and bring those products to market by co-developing the product with them. And I know, as you all know, that there is competition in flavors and fragrance industry. But what differentiates us from other companies are our patent-pending designer pathways that give high yield and our ability to rapidly expand our portfolio. We are also a products company. We don't offer any research services. We are laser-focused on our portfolio, and we bring those to market rapidly. In the recent conversations we had with our potential partner, they mentioned, and I quote, "There are very few synthetic biology companies of your size and at your stage who can hold up a product in their hands, and that's what makes you different." And I say we don't hold up just one product, we can hold up 3. Our team currently consists of myself and Jonas Miller. Between two of us, we have 20 years of experience in metabolic engineering, bioprocessing, and biochemistry. And we have a team of expert advisors who are guiding us to de-risk not only just the products, but also commercialization of our technology. We are Team Ardra. We are raising $1 million in seed round to bring our first fragrance molecule to commercial scale and reach early revenues. And if you would like to know more about us or missed out on the chance of smelling the fragrance product, I request you do come to our booth. Thank you.
Hi, my name is Charlie Ye, CEO of MPholytics. Our mission is to fight epidemics with rapid molecular diagnostics. Several weeks ago, we sent our co-founder to Brazil, to the epicenter of Zika outbreak. What she found was shocking. Within one single lab, there were 3 ice chests like this, full of thousands of backlogged patients waiting to be tested for Zika. Most of these samples will not be tested. Even if they are tested, it will take weeks or months for results to get back, which will not make a difference for the patients or for outbreak containment. Without rapid, accessible diagnostics to determine who is infected, we cannot implement control measures properly. And this is exactly what happened with Zika. Within a year, it spread like wildfire. The scary part is Zika is not the first, nor is it the last infectious disease to hit us. Within the last century, we had hundreds of outbreaks, most of them concentrated within this decade. We had avian flu, influenza, SARS, Ebola, and now Zika. With modern air travel and rapidly evolving pathogens, epidemics continue to spread. Unfortunately, current solutions are not adequate. Dipstick assays, similar to your pregnancy test, are not sensitive enough to look at early detection. On the other end of the spectrum, PCR, highly sensitive, but it's not portable, and it is also very expensive. Costs 10 to 100K. Because you can't bring PCR near a patient, it can take weeks to get the results back. Isothermal amplification is also as sensitive as PCR, but it's also costly. We need a new solution that is low cost and sensitive that could be brought to the field. Our solution is a fast chip. It is so small that you can literally put it in your pocket and bring it out to the field, the fast chip. This chip was originally funded by the Gates Foundation and DARPA with the aim to bring the lab into the field. It is fast. Within 30 minutes, we can test for results. Patients no longer have to wait for days and weeks to get the results back from centralized labs. It is fast. F stands for field ready. The entire system is portable. The user simply opens a vacuum seal pouch, loads the sample onto the chip. puts the chip onto a heat pack for 30 minutes, then puts the chip into a reader, and then uses a smartphone to take an endpoint result. What sets us apart from all the other microfluidic technologies is our core vacuum-based fluidic actuation technology. Without using any external equipment, pumps, power sources, controllers, computers, we can run this chip. This is why this chip is so small and ultra low cost and ready to be brought to the field. It is affordable. We plan to use injection molding to make these chips and readers. We estimate at scaled production, it will be orders of magnitude lower cost than the current PCR solution. Sensitive. We are using isothermal nucleic acid amplification to test for DNA and RNA. Isothermal amplification has comparable sensitivity to PCR, which is the gold standard. We can run quantitative tests on this chip, useful for applications such as HIV viral load monitoring. For example, the more green dots you see, the higher pathogen concentration in your blood. As another mode of using this chip, we plan to run hundreds of simultaneous reactions on this chip and screen for multiple diseases in one single chip. For example, if you have cold symptoms, one chip will tell you which kind of bacterial or viral infection this is, so you can have proper treatment. Trackable. We use a smartphone app to analyze the results, aggregate the results, the GPS coordinates, the patient data into a cloud-based tracking system. This becomes a very powerful disease tracking platform. Ministries of health can use this data to determine where they should put their resources first in the hotspot. Our go-to-market strategy is tiered into 3 stages. Initially, we'll be selling this as a research tool. First, as a PCR alternative, then sample prep module. After we integrate these 2 together, and after we get FDA approval, we'll market this as a point-of-care diagnostic assay for humans. To validate the market, we've talked to OBGYNs, local doctors, hospitals, and researchers, and they all express strong interest for this technology. There's also interest in mosquito surveillance to test mosquitoes for dengue and Zika to track diseases. We have licensed 4 patents from UC Berkeley. These patents cover the fluid actuation, the overall system design, and the patterning on chip. We have a strong founding team. Dankar, our co-founder, was the original lead inventor of the TB Cepheid assay, which became one of the world's leading point-of-care assays. I was the original inventor of the fast chip, which is funded by Gates and DARPA. We are working with world-class collaborators, the first scientists to discover Zika in Brazil. Here's a video of our field study.
So we need to develop this technology before the next outbreak so that we're prepared to go right into the field and contain the outbreak.
Tiene que ser un diagnóstico rápido, sensible y barato. Es importantísimo para evitar la diseminación de la doença.
We've developed a test to diagnose people anywhere in the field, in a clinic, in their homes. It doesn't need any power. Do molecular detection of DNA or RNA of different infectious diseases.
Esse equipamento não precisa de técnicos super treinados. É a eficiência do teste e sua alta qualidade. Estamos todos satisfeitos com os resultados encontrados.
We need to make sure that this project gets off the ground and that it works properly and as expected with high sensitivity and specificity in all situations. And so we are asking for funding so that we can continue this work.
As you can see, there's a huge need and huge market for diagnostics. But why are we personally motivated to do this? Danica has worked in the field and experienced firsthand the heavy cost of delayed response. As for me, even at home, when I go to the doctor, I like to see my results right then and there and not be anxious about it. Imagine how much more Zika patients must anguish while waiting for their backlog results. We have technology in our hands that can change how diagnostics is done and save millions of lives. We invite you to join us in this mission and fight Zika and the next 100 outbreaks. Thank you.
Your genome, can everyone hear me? Okay. Your genome provides information about every biomolecule that is produced in your body. As such, it is a linchpin in guiding healthcare that is tailored to you in this incoming era of precision medicine. I'm Carlos Araya, the CEO of Joongla, where our mission is to accelerate the insights that we all stand to gain from understanding our genomes. We do this simply because whether we're considering a mastectomy, a defibrillator implant, or a targeted anti-cancer therapy, we rely on our genomes at many of the most critical moments of our lives. And although there have been remarkable advances in technology, some of the fastest in the history of mankind, these technologies that have brought personal genomes to the clinic, we find ourselves today in the middle of a plague of uncertainty because sequencing is not understanding. And the reality is that we know very little. In fact, today, we know the effects of only 0.1% of the possible variants in disease-associated genes. And yet, this limited knowledge drives what is a $6 billion genetic testing industry. And although we can acquire sequence information very quickly now and cheaply, the reality is that the knowledge that we rely on here grows painfully slowly. For example, in the last 6 months, we've uncovered only the effects of a minuscule fraction of the variants in BRCA1, one of the genes in which we seek actively for these associations. So rather than waiting, at Joongla, we believe that there is a better approach, which is to lens the knowledge that we have today to shed light across this landscape. That is, to couple genomics, biophysics, and machine learning to understand and predict the effects of variants. To do this, we first need to recognize that variants affect molecular functions and not just sequences. And while it's easy to obtain sequence information today and to associate those individual variants with genes and proteins, what we really seek to understand is how each variant impacts a specific molecular function, such as ligand binding in this example. So what we really need to enable is a new understanding of the genome. At Joongla, we've developed methods that allow us to measure functional relationships between different parts of the genome. And with these, we can then take the knowledge that we have today and pinpoint how disease-associated mutations are affecting specific functional modules, such that when we see new variants in the clinic that affect these modules, we can infer these are likely pathogenic. We marry these proprietary insights with machine learning into software that enhances existing genetic tests. Our products are gene and disease-specific interpretation engines that provide doctors and patients with accurate, comprehensive diagnostics that are supported by robust performance metrics. We've shown our software increases the performance of even the best genetic tests in the market. It does so by increasing the ability to identify patients who are at risk of disease without mischaracterizing patients who are not at risk, as is shown here for BRCA1. But this is not a one-trick pony. In fact, we've shown that this approach improves the interpretation in 94% of the genes that we've tested, yielding, most commonly, a 28% improvement in the accuracy of each test. This opens up large market opportunities because these analytic solutions can be added to existing tests without the requirement for FDA approval. and therefore they can be used to enhance the accuracy of the 60,000 genetic tests that are in the market today. Over the last months, we generated solutions for 15 of the highest volume genetic tests in the market, including 8 genes that are recommended for universal screening, and in which we can therefore expect over 100 million patients will be analyzed over the next 10 years. We know today that some of these genes generate revenues of roughly $600 million for individual providers. But with sequencing becoming a commodity and patients walking into the clinic with their genomes already sequenced, we believe that the value in genetic testing is increasingly in the interpretation. So our tiered go-to-market and revenue strategy begins by leveraging our multiple patents to deliver our insights as a service, to then improve existing genetic tests, and to then integrate clinical data to generate new tests, such that we become a global distributor of genomic insights. And although I cannot disclose names, we're today working on partnerships in each one of these phases, including with one of the largest sequencing companies, one of the leading genetic test providers, and one of the largest heart hospitals in the United States. In our business model, we begin by leveraging public data to generate gene and disease-specific interpretation engines that we then distribute to existing genetic test providers and collect revenue on through volume-based licensing, following a software-as-a-service model. But in addition, Joongla partners with hospitals to generate new companion diagnostic tests that accompany drug prescriptions. And although these types of tests do require FDA approval, they can each be worth as much as $100 million pre-market, which then well justifies the investment in the clinical trials where the clinical data is strong. So we view these really as partnerships. And Joongla partners with these hospitals, incentivizing them to provide data by giving them a new revenue stream through royalties. And this is important to the mission of Joongla, because whereas most genomic... This might be running low on battery. Whereas most genomic software companies today are focused on collecting sequencing data and aggregating existing knowledge, Joongla is among the few companies that are generating new knowledge for distribution across the industry. Our competitors are companies that are either using low-throughput versions of technologies that we've invented or are companies that we've done due diligence for. In either case, we understand well their practical and technical limitations. We're a team of Stanford scientists with over 15 years of experience in genomics and biophysics. Our work has been published in nearly all top-tier journals and featured in the New York Times. We're supported by leading experts in molecular sciences and genomics, including Doug Fowler and Mike Snyder from Stanford University, and are part of the leading biotech accelerators. Our goals are to complete our multiple validation pilots and reach first revenue within our first year. To then focus on capturing 15% of the total addressable market for a specific genetic test by 2018, and to be ready to initiate a first clinical trial shortly thereafter for a companion diagnostic. To reach these goals, we're raising a $2 million seed, so contact us to bring accuracy and new capabilities to the genetic test that we all rely on. Thank you.
All right, great crowd.
My name is Morteza Ahmadi. I'm the founder and CEO of Kidney Labs. We're developing an implantable artificial kidney for patients with end-stage renal disease. There are 650,000 patients with end-stage renal disease in the United States, and there are only 20,000 kidneys available for transplantation each year. The demand for kidney is so high and the number of donors is so low that patients sometimes have to wait 5 to 7 years to find a kidney for transplantation. The only option they have to use to survive in those years is dialysis. Dialysis was invented by Dr. Kolff in 1943, and it has been saving lives since then. However, the technology has not changed much for decades. Imagine yourself attached to a huge dialysis machine, watching your own blood in circulation in plastic tubes 3 times a week for 4 hours each time, with not much hope for any sort of change in near future. Such patients are in pain, and they suffer emotionally and physically. In fact, the mortality rate of patients under dialysis is 65% in 5 years. And dialysis costs about $82,000 per patient per year. And that makes dialysis a huge market. Dialysis was valued at $70 billion in 2015, and it's estimated to grow to $100 billion by 2020. My journey was started at Kidney Foundation of Canada when I was a fellow and I was developing a miniaturized blood dialyzer based on silicon nanotechnology. Silicon nanofilters were used by leading scientists around the world to build artificial kidneys. But to our surprise, the technology failed in material performance. Since then, I have been on this journey to build a reliable technology to build artificial kidneys. And now we have it. We have developed a unique, reliable, hemocompatible technology which mimics the filtration properties of kidneys. And now we tested that in the lab, and the result was amazing. We then packaged our technology in a biocompatible casing, and we built a device. And here is an implantable artificial kidney, and this is the prototype here. This is how our device works. Our device is connected to a major artery. The blood is diverted from the artery into a nanofiltration system, and the nanofiltration system removes uremic toxins and fluids from blood. The waste products go to bladder and are removed like a normal person. Blood is then passed back to the vein. The level of uremic toxins and fluid in the body of dialysis patient peaks 3 times a week, and that's mainly before the dialysis session. Our vision in Kidney Labs is to keep the level of uremic toxins and fluids at a normal and safe level at all times using our device continuously and automatically. We were confident about our results in the lab that we decided to do animal studies with our device. To do that, we first wrote a protocol and submitted that to Ethics Approval Committee at UCLA, and we did our study in a facility affiliated with them. After receiving the approval, we went ahead with the study, and we implanted our device in 2 pig animal models with no renal function. In the next slide, I'm going to take you directly to the surgery room. And I'm going to show you how our device functions in the surgery on the living pig.
Oh, just another one.
Oh my goodness.
Wow. That's the fluid that we removed from the living animal. We work closely with clinicians at Stanford University, UCLA, University of California at Irvine in the United States, as well as Grand River Hospital and St. Joseph's Healthcare in Canada. Our FDA pathway is PMA, and in the next 2 years, we make sure that we do enough tests in animal models that the device is safe enough to be used in human. We then follow up that with our clinical studies, phase 1 to phase 3. Our plan is to bring the device to market in 6 years. Our business model is based on selling our device to hospitals for $100,000 with 65% profit margin. The hospital will be reimbursed from health insurance for $200,000, and the patients will be receiving the device for free. In comparison with the current technology, which is dialysis, we have several advantages. Our device provides treatment continuously and automatically, which provides more freedom and a more normal life to patients with end-stage renal disease. Our intellectual property covers the technology behind our nanofiltration system, as well as the technology behind our device. All this have not been possible without my team. Our team has expertise in biomedical engineering, nanofiltration, and management science. In our advisory board, we have key opinion leaders in surgery, nephrology, and expertise from people who have brought implantable devices to market. We are seeking to raise $2 million in seed round, and we want to use that to de-risk the technology to a point that is safe enough that we can use that in human. Thank you very much.
Well, thank you. How's everyone feeling? Was that, well, I hope that wasn't too long. Thanks. Yeah, really a big round of applause for all the team. These are scientists that aren't used to doing this. I mean, this is incredible to watch the stage fright. So. It's remarkable to see the transformation in a lot of these scientists. I mean, going from an insight to having to deal with business, to having to deal with talking about themselves in a way they've never talked about before, right? Like, you know, not being so humble that you're not going to say anything at all. It's completely different and remarkable to see, again, the transformation that you saw on the stage. So thank you all for being part of that. That means a lot to me and the entire team here at IndieBio. Why don't you guys come on up? So there's a couple things, housekeeping before I want to introduce you to a couple other people. But one, so for the first hour, we want to give the teams here access to investors and press preferentially. Sorry. But that's one of the big reasons they're here. So we're going to have some, what do you call it? Those stanchions where we'll separate the crowd, but after an hour, we'll open it up to the entire crowd, of course, and that's just with the CEO. So the teams will have some of their other team members come out, and we could all meet them. If you're going to taste some food, taste Miraculics last, because that Miraculin does have 30 minutes of amazing taste, and so it'll affect the other ones. So I want to make sure that you get the real deal for every single team. So with that, I would like to introduce you to one of the most remarkable men I've ever met, Sean O'Sullivan. When I first met him, I talked about biology and its changing impact in the field, and he said, yeah, that sounds like something we should do. Very few people on the planet are willing to do that. So I'd like to introduce Sean to say a couple things about what you just saw.
Thanks. Thank you very much. I mean, so I'm Sean O'Sullivan from SOSV, IndieBio, like our other family of accelerators, Hacks. Arvind is a general partner at SOSV as well as myself, et cetera. I'm the managing general partner at SOSV. But anyway, so I kind of feel like we should do a quick test to see if everyone remembered all of the biology that you've just learned. I feel like anyone who attended this should basically get a bachelor's of science in biotech or something, because there's so much that's going on, and actually so much on the cutting edge of life and discovery. And that has happened in these. We keep talking about, oh, IndieBio, it's only 4 or 5 months. But there's quite a few months that goes into even selecting these companies to get into these programs. But more importantly than that, there's the 15 or 20 years, or really the whole lifetime that people have spent developing their brains to the point where they really know the engineering, the science, the math that makes all of this possible. So I just wanted to say I'm honored to be part of the incredible revolutions that we're seeing here today. I think it's remarkable that we had it was this week, right? The kidney implants in a pig. It was Monday of this week. First time in history, right? And that's an advance, hopefully, for mankind, right? And that happened as a result of this work that that the team obviously did, that pulled together, but enabled because science is now being enabled to accelerate into commercial companies through this accelerator, IndieBio, leading this effort. And I'm so proud of this entire team, and I'm proud of you guys for coming and supporting these teams. I hope you guys have big checkbooks so you can support these companies, because there's some incredible work for humanity and for the quality of life and the environment that all of these companies represent. So I hope you really take your role as investors and as human beings seriously by really considering these companies to the greatest extent that you can, because they are really transformative. It's not just another messaging app, when you're coming to IndieBio. So without further ado, I guess, what do? We just let the story go on.
Yeah, thanks again for coming, and please hang out. The bar is open, and investors and press, please come over, and thanks again.